Today we will start somatic embryogenesis. Somatic, soma means tissue. Embryogenesis, that already been taught to you. Now somatic embryogenesis is also a tissue culture techniques that involves regeneration of plant from somatic cells. Somatic cells will generate a plant. Now somatic embryogenesis, as I said previously, same things, same words I am repeating. Somatic embryogenesis is the process which is involved in plant tissue culture where somatic cell, remember these, that which are non-reproductive cell of the plant. Non-reproductive means it is basically formed from the tissues, undergo series of developmental changes, series of undergo that developmental changes. Now the word change. Up to this we are dealing with the somatic tissues. To form embryos without need for fertilization. Embryo is there, but to form embryos without need for fertilization. This means these This means that all new plant can be generated from single somatic cells. You are utilizing only a somatic cells of single one. That now remember these words. When we are writing a somatic embryogenesis definition of that, these word be, it will be removed, reproductive parts. Means we are bypassing the normal sex reproduction process involving two things, pollens and ovules. We are not involving any things regarding sexual reproduction process. We are only utilizing single somatic cell for culture. And this first reported somatic embryogenesis in Daucus carota by this Re-Inot, J. Re-Miller. They have shown that the capability of somatic plant cells of a culture to produce embryos, and that is known as embryogenic potential of that particular plant. Every somatic cells have a ability to produce embryos, but this is not general for all the plant tissue culture techniques or all plant species, depending upon. Today in human being also, they are utilizing techniques we are called as somatic tissue cells. Okay. So the development of pathways for numerous well-organized small embryos, now embryos, these to produce embryo, resembling, that is zygotic embryo. Zygotic embryo means involvement of sexual pollen and ovule, male and female gametes. From the embryogenic potential to somatic cell of callus tissue of suspension culture is known as somatic embryogenesis. And that is, as I said previously, that has a capability of somatic cells of culture to produce embryos. So this is all about the introductory parts, or you can say how the embryogenesis can possible. Now, what is the principle involved in this embryoid production without somatic cell, without reproductive cells? So, principle, it can be possible with the two ways, or you can say two things can be possible. So, how this possible? One is direct embryogenesis, that will not covered in here. So, I will give you this. Direct embryogenesis and indirect embryogenesis. This is the principle through which you can produce somatic embryogenesis process. Now, direct embryogenesis means pre-embryogenic determined cell. You are assuming the fate, you are assuming the past of that or future of that predetermined fate to form embryo and await some kind of stimulation from embryos. Now, indirect embryogenesis, or you can say IDC, that is induced embryogenic determined cells. That can be asked in the full form IEDC and PEDC. PEDC means pre-embryogenic determined cells. IEDC means induced embryogenic determined cells, and both these phases are involved in direct embryogenesis or indirect embryogenesis. Sometimes they are also asked what is the difference between direct embryogenesis and indirect embryogenesis. So somatic embryogenesis, as I said previously, that is initiated in two different phases. One is direct, another is indirect. In some cultures or culture techniques, somatic embryogenesis is initiated by the pre-embryogenic determined cells, that is PDCs, and in PDCs, the embryogenic pathways is predetermined, and the cell appear to only wait for synthesis of inducer. So how it is occur when embryo are started directly from the explant. Explants mean maybe a leaf, maybe any tissues from up to stem. Started directly from the explant, creating identical clones. In other words, without callus formation of embryo from the explant, without the callus formation from the explant, that is called embryogenesis. Now, ID, direct embryogenesis, indirect embryogenesis. As I said, direct embryogenesis occurs when embryos are started directly from the explant, creating and identical clones. In other words, without the callus formation of the embryo from the explant, that is called direct embryogenesis. Now, indirect, or you can say indirect development or direct embryogenesis, occurs when explant produce undifferentiated or partially differentiated cell, often referred to as a callus. Here callus is involved, IDE, which that is maintained or differentiated into plant tissues such as leaf, stem, or roots. For example, 2,4-D, 2,4-dichlorophenolic acetic acid, 2,4-D, and BAP. This is the two media composition in which they are AD, 2,4-D and BAP, for indirect embryogenesis. So, benzyl aminopurine, BAP, and gibberellic acid has been used for development of indirect embryo in the strawberry also. So this is the direct embryogenesis and indirect embryogenesis. I will give you a more explanation of ID. So indirect, in some cases, somatic embryo development needs some kind of callus formation, and embryos emerges from indirect embryogenic determined cells. Now, indirect embryogenic determined cells that require or redetermination to embryogenic state by exposure to specific growth regulators. As I said previously, that is 2,4-D. In most of the cases, indirect embryogenesis occurs where it is induced under in vitro condition. So, I can say two different BD are required. One for initiation of embryogenic cells, that induction medium. We require induction medium. This induction medium must contain auxin. Remember this word. The induction medium must contain auxin to initiate somatic embryogenesis. It is removed or reduced to minimal level in second medium. First medium and second medium. In second medium, which support the subsequent development of this cell into embryos. Embryos are generally initiated in the callus tissue from superficial clumps of cell that is associated with the large vacuolated cell and that do not take part in the embryogenesis. Occurs when embryo are started directly from the explant creating identical clones. As I said previously, the definition of embryogenesis that without the callus formation of the embryo from explant. Now, embryogenic cells are characterized. How it is characterized? How it's look like? Embryonic cells are characterized by dense cytoplasmic content. Dense cytoplasmic content. For example, large starch grain and relatively large nuclei with darkly stained nucleus. This is the primary characteristic through which embryonic cells are characterized. Dense cytoplasmic content, large starch grains, and large nucleus. Each developing embryo passes through three sequential stages of embryo development. How any plant tissues. In physiology, they are giving you a differentiation. If you studied in the plant tissue culture techniques, they are also give how the shape is changed under the microscope. When callus formation or you are dealing with the embryo or embryogenesis. So there are three stages of embryo development. GHT. There are a large number that I will explain you and showing also. Globular, name itself globular. Heart shape, name itself heart. And third is torpedo. Torpedo stage is a dipolar structure which is ultimately give rise to complete plantlets. Which is ultimately give rise to plantlets. Now uses of somatic embryogenesis. Up to this, you have to remember only what is direct embryogenesis, indirect embryogenesis, embryogenic development stages of embryo development. Then what is the application, what is the significance, and what is the use of somatic embryogenesis. As I said previously, this word is somatic embryogenesis. We are dealing with the embryogenesis but without the callus formation. So plant transformation techniques that can apply for somatic embryogenesis. Another is mass propagations. You want to create a, you have only a leaf tissues and you want to create a large number of same population, same clones with smaller tissue. You can develop with somatic embryogenesis. Now I will show you the schematic or figure representation of somatic embryogenesis in the carrot. I am giving you because of this VSC course part. Both are comes in your. So you have to put a floriculture somatic embryogenesis in your slide. So first of all, we should know how these A, B, C, D, E, F and G formation up to in the tissue culture techniques we are dealing with the somatic embryogenesis. Any explant, you have 9 crops. Out of them, any plants you can take as a leaf tissue as an explant. Okay. Then you are preparing a media, macromedia and micromedia. After that, solidified in the petri plate or culture tubes. Then, after solidifying, preparing media, you are putting first sterilizing that leaf tissue. After sterilization, cut and put into 3-4 parts in the same petri plate with then putting for certain period of time. It may be a 6 days, 4 days, 7 days, 8 days, depending upon your plant tissues. That will gives you a callus formation. This callus induction you have to see under microscope. As I said, in embryogenic, somatic embryogenic stages, first is H, G, H, T, heart shape, globular shape, heart shape, and torpedo. Now, globular embryo you are seeing in the under microscope, then you will see a heart shape. Sometimes explant is also put into a suspension culture also, depending upon the protocols. Then you will find a globular embryo or heart-shaped embryo. Same thing is with here also self suspension culture is there, liquid culture is there. You will find a globular embryo. This globular embryo, another stage for callus or somatic embryogenesis is heart-shaped embryo. It's look like this. And finally, the mature embryo that is called torpedo stage. Torpedo have two polar regions. So you will see under microscope like this. And finally, the regenerated plantlets. Before that, you have to give a shoot formation and root formation, both stages. And that intact plant you are giving in a with thermocol with the Hoagland solution for the hardening or removing that solution for 1 or 2 days, and then you are putting into a soil media, first soilless media and the solvent soil media, and then hardening in a greenhouse, or if it is possible then you can put into a plastic also. And so where is the application? As I said, that will give you a mass culture population, micropropagation with the number of plants you can multiply with the leaf tissues. Now, somatic embryogenesis will possibly you will find in the vegetable crop, flower crops, or our crop tissues. Many but tree species also can be fruits possible, but no one has tried that because rapid multiplication of elite cultivars, that is application of somatic embryogenesis. Your course is floriculture. Here I have taken the PPT of VSC. Therefore, it is given an example of vegetable crop improvement. But you have to write a floriculture, flower crop improvement. So, somatic embryogenesis, elite, rapid multiplication of elite cultivars. If you have single or a very small germplasm, you can multiply number of trays you can produce with that, only a one single cell. Or you can say somatic embryogenesis can be propagate elite cultivars, and this technique allow production of large number of. Now remember this. These word you have to write. Elite cultivar multiplication you are only when. But what it means? It means genetically identical clones or genetically identical plants you can produce in very short period. That continuously high quality planting materials can be produced with the rapid multiplication of elite cultivars. So one of the ways of application of somatic embryogenesis. Now another things, it can also, this is universal for all plant tissue culture techniques. This application or advantage, most of the same words you will find in every protocols. So enhancing disease resistance. If your explant is free from disease, otherwise it is not possible. So somatic embryogenesis, why it is good? In most of the very reproductive, for example, pollen or your ovule culture, that is mostly very, you can say very smooth, or we can use word, it is very delicate. So anyone can attack on that issues, softening of that. But in case of somatic tissues, that hardening is there, wax on the upper surface is there. So the attack of most of the pathogen cannot be possible. So somatic embryogenesis can be used to develop a disease-resistant variety of flower crops or vegetable crops by inducing callus formation and from the somatic cell, regeneration of hybrids. Many people are telling, but not like this, as we are doing in the field. Regeneration of hybrids. I will give you in protoplast fusion or somatic hybridization. Regeneration of hybrids. Somatic embryogenesis can be used to regenerate hybrids. How? Which are often difficult to propagate by traditional methods. That techniques can be used for large number of hybrid productions, or you can produce genetically uniform, as I said previously, and have a desirable traits. Now, I have put in one words genetic engineering, but it is not that much suitable. But why I am giving here? Because in every protocols of genetic engineering, you must have a knowledge of plant tissue cultures or a micropropagation techniques. Otherwise, you cannot transfer any genes. You should, you must have a knowledge of plant tissue culture. Therefore, I have put in one of the advantages in the somatic embryogenesis, application of somatic embryogenesis. If you want to transform any gene with the agrobacterium mediated genetic transformation, you have to prepare a culture in the flask, then infected with the tissues, and as I said previously, the same stages you have to follow. Now, in this, introduce desirable traits into a flower crop or a vegetable crop through the genetic engineering. This technique enables precise manipulation of the plant genome and creation of plant with improved characteristic. That may be a abiotic stress resistance, or you can increase the nutritional value. As I said previously, that is gene insertion with direct method, gene gun, or conserved of endangered species. That is also one of the advantages of plant tissue culture. So somatic embryogenesis can be used to conserve endangered species, then preserve genetic resources or genetic diversity of that plant. Then, in summary or in a conclusion, you can say somatic embryogenesis is a powerful tools that can improve the flower crops by enhancing their characteristic, improve their resistance to disease and pest, and preserving their genetic diversities. So we will start with the somacloral variation up to that direct, indirect embryogenesis and what is their application. For application, I have already described. Somaclonal variation. Soma means tissue, as I say. But here is the genetic variation that occurs in the plant generated through tissue culture. Soma means tissue. Variation in the tissue culture. Then soma clonal variation. Clone. Variation. Soma clonal is the genetic variation that occur in the plant regenerated through the tissue culture. Due to genetic changes that happen during regeneration of tissue culture techniques, or you can say a tissue from the culture cell. That changes can be in the form of chromosomal rearrangement. Now, these words you have to remember. As I said previously, so chromosomal rearrangement, mutations, or epigenetic modification. What is somaclonal variation? Somaclonal variation is the variation happens during the plant tissue culture techniques or that maybe the changes can be formed with three words. First, chromosomal rearrangement, or you can say mutations, or epigenetic modification. Somoclonal variation, as I said, in every tissue, plant tissue culture, it is useful for the plant variety development. Same words you have to repeat. So it helps in the identification and selection of superior plant with desirable traits such as disease resistance, enhanced nutritional quality, and improved stress tolerance, or increase the yield. If you have a proper marker for that, otherwise you cannot with only a tissue culture you can say that this variation, this chromosomal rearrangement is there, that higher, this rearrangement is for. But you should have a morphological data before that. You cannot compare that these chromosomal variation or these chromosome changes are there or this mutation is there that will give you a higher yield. But you have to prove, or you should have certain primary data for that. So this is in general. I am giving you an explanation. You know very well everything you know. What is somaclonal variation? What is indirect or direct embryogenesis? This is all are repeat. Application of somaclonal variation. There are certain stages. So first step, regeneration of plantlet through plant tissue cultures. As I said, plantlet from single tissues or single cell you can or leaf tissues. This can be done by growing tissue explant in the culture medium. As I said, every protocol requires a different hormones and different composition nutrient medium. So depending upon crop species, you require to set a protocol for plant tissue culture. So first of all, you should have this protocol, or you can generate also. Screening for desirable traits. Now, first application of somoclonal variation. As I said, the tissue variation you can see under the microscope also. Screening for desirable trait, regenerated plant are screened for desirable traits. As I said, drought or salinity, abiotic stress. How this is possible in the tissue culture? For example, you have taken an explant of our crop, solenesis, or you have a rose plant, or any. You are putting a tissue into a various medium with higher salt concentration, zero point 0.4, zero point 0.6, zero point 0.8, depending upon the change in the pH as well as change in the salinity you are putting in the medium, and then generate a killers. Whether the you are putting a 1% salt in that culture media, what is happening? Whether the killers is generating or not. If it is not generating, means it is susceptible to salinity. So screening of desirable traits can be possible in the plant tissue or somaclonal division. Then selection of promising plant with desirable characteristic are selected and propagated vegetatively to ensure the desirable trait are stable. Now selection of promising plant, how you can generate? There are certain, after the generation of shoot formation, you can see the structure of leaf also, morphological data, through which you can select this plant in this condition is survive. Therefore, we are choosing this. But after the generation or you can say after the hardening, it is very difficult to select. But in application of somaclonal variation, we are putting this. Then third, evolution and testing. Selected plant can be evaluated in the field trials and perform if it is well under different environmental conditions. Then you can select. So the first stage, as I see it, up to that, one words direct embryogenesis and indirect embryogenesis, somaclonal variations, now organogenesis. You have to write only a words, and you these words I will provide you a material for that also. Okay. So organogenesis, as I word itself, organ and genesis means formation. So development of adventitious organ or primordia from undifferentiated cell mass in tissue culture by the process of differentiation. Same you can see in the callus culture formation up to here, and then after the shoot generation, this shoot cut into a rooting medium. Okay, this procedure is followed in all the culture techniques, but it requires a different nutrient medium for different explants. So when peripherally cells start dividing in the normal growth, it will degenerate after some time. Some abnormal structure, which are structurally similar to organ, but between the dermal, vascular and ground tissue present in the plant. The true organ, because they are obtained directly from the peripheral cell of the callus. So here you will not find as such shoot formation. Callus you are eating, that will form like this. You are cutting, the more number of shoot is there, one leaf is coming, you are cutting that and putting in transfer media. Similarly, not organized meristemoids or patches of actively dividing cell of the callus you will find the organogenesis. Now, very important for tissue culture techniques in every stage or every culture techniques. This is as such factor affecting you can put into a plant tissue culture or organogenesis. So following factors, you can say one is size of the explant, or you can say choosing of the explant, ages of the explant, shape of the explant is also affected, but generally they are not putting in these. Younger leaves produce roots. Remember this word. This is universal truth. Ages of the explant is very important for choosing a first explant in tissue culture. If you choose a younger leaves, purely mature younger leaf, that will certainly give you a root tissue. If you take older leaves, that produce root after organogenesis. Season is very important for choosing the explant. In which season you are choosing? Mango. Annually you will find a various leaf tissues. Seasonal explant. Explant excised during summer or winter do not produce root at all. Now words you are confusing. Sir, these two seasons is very important for us. How you can say that summer and winter? And which season we have to take? If you are only at the monsoon is remain. But during monsoon, there are a number of factors affecting. For example, the contamination is higher possibly during the monsoon season. Why monsoon is important? When you are choosing the explant or leaf tissues, they will generate very rapidly during that monsoon season. God has created that every plant is rejuvenate, most of the plant in monsoon season. Therefore, if you choose this season, that will give you higher growth in control condition. So eggplant exercise during the summer and winter do not produce shoot at all. But you can produce during the summer also and winter also if you choose. That doesn't have any home rule that you can in autumn season you are collecting, winter season you are collecting. But for the selection of seasonal, there are three things for choosing the eggplant. Now tell me. First is
size of explant, then age of particular explant, and last one is the seasonal explant.
season of the eggplant. Now two things. One is calogenesis. Another, kalo means callus related with it. Rhizo means rhizosphere like. So oxygen gradient is you will find low in calogenesis. Remember this word. I will ask in the match the following, or someone also ask in the multiple choice also. So calogenesis related with low, you can write also. Rhizogenesis related with the high oxygen gradient. So oxygen gradient high and low. Low with calogenesis, coulogenesis you can say. Calogenesis, and another is rhizogenesis. Now fifth factors, the intensity of light, or one can say the color spectrum of the light is also affected in the eggplant also, where you are putting how much light you are giving to that. So blue spectrum or blue color of spectrum and red color of spectrum. Generally, blue color of the spectrum are useful for a production, or you can say the organogenesis formation. They are utilizing blue color spectrum. But as per the research, and many researchers have found that the red color intensity promote routines. These are the key points of any plant tissue culture techniques. And you can ask from your student also. Biotechnology, elementary plant biotechnology. Have you heard some? Plant tissue culture? Yes. What is the care you should take during the selection of the or choosing of the plant? How much light is required? How much lux is required for that? What is the medium you require for that? How you are transferring the plant from one media to another? What care should be taken? That is not written. Practically, you should understand. When you go for practical, you will understand what are the problems created during that. That is not given in any books or even research paper also. They are not giving you, I have put it these excellent because of these, and these are the reason for not generation of. They are simply giving the transformation that many percent and regeneration of plant that many percent from these excellent. Or I have utilized that many concentration of hormones and explant is generated that much. But they are not giving you the reason for that. So blue color of spectrum and red. So red color promotes routine. Light durations universally 16 hours and light intensity is 2000 to 3000 lux is compulsory required for any any plant tissue culture protocols. Certain gives 8 hours, 12 hours, 16 hours, but 8 to 16 that is universal for all protocols. And light intensity is 2000 to 3000 lux and temperature is 20. In many research paper, you will not find these hours, lux and degree temperature. They are giving only, as I said, percent. of
Survival.
plant regenerator or survival. Ploidy level, that therefore you require some cytogenetic study, or you can say cytogenetic techniques you require for determination of ploidy level, whether it is change or not due to hormones or due to these various techniques. So factor affecting organogenesis I have already discussed, and giving a very synthetic seed production we will not take.